I'm going to lead off. I'm Rebecca Wattam. I'm part of the Cyber Infrastructure Division. And we've had a long history of providing science portals, analysis, and data integration. And Chin-Hong and Andrew are also going to speak about what we do today. Uh-oh.
Forward button on the keyboard.
Oh, okay. That's even better. Or the down button. Right now we have three projects that four, that we have going at PATRIC. In cyber infrastructure, the first is the all bacterial and now all archaeal genome project, which is called Resource Center, which is PATRIC. We also have an RNA-seq analysis portal. We have some new developments in pan-genome analysis that we're going to talk about today, and also the genomic and proteomic architecture of atherosclerosis, which is a really hard word to say, which is GPAA. And so we're going to discuss, different speakers are going to talk about that today. The thing that unites us all is, in our group, is thinking about things from multiple perspectives of data integrating and providing analysis equipment. And one of the ways is you have to serve the computer scientists, which at VBI is most everybody here, and providing a way for them to integrate with the data is so easy because all they need is a command line interface. But I'm from a different perspective. I'm a biologist. Did you ever read that Dr. Seuss book, The Lorax? Remember, the Lorax spoke for the trees? I speak for the biologists. And the biologists, when you think about it, all the information, they have all the lab information and all the knowledge, but they are often lacking in this ability to be able to do things computationally. And so part of what we do is try to develop things so that to help them do their own research. And so we design different interfaces to help them. Now, they can always ask us for analysis help, but that's, sometimes, or they hire people like the DAC to do the analysis. But the fastest way for them to gain new insights is to provide them with tools that they can use and understand to get that data and interpret it themselves. So that's part of what we're all about. So lately, I've talked to you guys about PATRIC a lot in the past and how we've gone from 8 genomes to now more than 40,000 genomes, and how we have transcriptomic data and host pathogen interactions, everything embedded there. But we're also very responsive to new calls, and lately NIAID and NIH has been extremely concerned with antimicrobial resistance. So in the past about 6 months, we've been putting a huge push in to try to meet that need and to bring in that data. Now, the way we bring in that data, there can be different ways that that data is brought in. Some of it is tagged to genomes, and I think this is going to be the most valuable method, is when you know that that genome is resistant or susceptible or intermediate to a whole antibiotic panel. If you can integrate that data so that it's easy for the biologist and also the computer scientist to see right away, then they can filter on that data and look at it. This is one of the methods we have. Now that we have this data, we have it's about 4,000 genomes from RNA, from antibiotic-resistant panels that we've brought in and tagged. Some of this has to be done manually because it's in the new phase, but now researchers can come in and grab all the data for resistance or susceptibility and analyze it that way. And so once you have this kind of tagging, and we try to bring in things from different resources. I know you had a question earlier about SRA or bringing in how do you get the different data. We try to bring that into PATRIC so it's just one link, one click away for the biologist. So we brought in a lot of antibiotic resistance data, but once you have that data, something like this, which is the TB drug resistant database, you can go mine that resource, which has a lot of detailed information from papers and individual experiments that look at specific nucleotide changes that result in antibiotic resistance. So you can take that and go into PATRIC and say, whereas these are done at a single genome level or in tens of genomes, you can now at PATRIC take that to thousands of genomes and say how many of them have this particular nucleotide change which is associated with resistant, which is this one, which is the valine that's highlighted there in the white. And so you can ask those questions, and now it takes what was known in the literature for a small number of genomes, then you can look more broadly and say, well, this generates resistance in rifampicin. Is that true? Can I look across a broader swath of genomes and see that? And you can also look, because these genomes are all tagged with different metadata characteristics like country of isolation or year of isolation, you could see that, oh, all of these are from Russia in 2010. And that's where we're seeing these resistant patterns. So that's the kind of things that we're trying to allow biologists the ability to do. Another thing, it was funny because we were on a call with some people at NIH who were really interested in antibiotic resistance, and the computational branch threw all this stuff at them, oh, you know, all the SNPs and this, that, and the other. But what they really wanted, what the biologists really wanted, was to look for presence and absence of antimicrobial genes across all the genomes. And we can do that so easily in PATRIC with just a heat map view, where black is the things that they're missing, and then the other colors are presence of more than one. So sometimes it's just a simple, something simple like this in a heat map view for the biologists to see to make the interpretations that they need. So now I'm going to pass off to Andrew. He's going to talk about RNA-seq and pangenome graph.